Hello, everybody. We're going to just wait another minute or two for everyone to show up. So just hang tight. Thank you. Okay, so I think it's time to get started here. So welcome everybody to today's webinar from the Protein Society, the Protein Science Special Tools Issue webinar. We have a distinguished panel today, and it's being led by Dr. Ben-Tal of Tel Aviv University. And we also thank Dr. April Rod of Wiley for organizing this. And so we're looking forward to this. I'm Chuck Sanders. I'm the president of the Protein Society. Let me begin by just making a few announcements about the society, and then I'll turn it over to Nir. So I want to remind everyone that our annual meeting is coming up July 7th through 10th in San Francisco. It's going to be fully in person this year. We're very excited about this. The abstracts deadline and the deadline for applying for travel grants has been extended. So we hope you'll get your abstracts and registrations in, and then we'll continue to take abstracts until the end of May. So anyways, we really hope that you can join us this summer. So our next webinar is Emerging Approaches in Membrane Protein Design, May 19th. And that also has a wonderful set of speakers. And so we hope that you can make that. And last, before I turn it over to the session, I want to point out that in a couple hours, our monthly Protein Society newsletter will be coming out via email. And there will be a special announcement in it this month. And so I hope that you'll take the time to open that, take a look at it, and read the other stuff in our newsletter. And so with that, let me turn it over to Dr. Ben-Tal.
Okay, so welcome everybody to this nice session. And as my new role as a senior editor in protein science, because of my computational background, I'm the natural person to handle the tools special issues. So I volunteered to do that. So from now on, I'll be handling it, and we are now preparing towards the 2023 webinar. Tool issue, and typically papers in the tool issues should be freely available. The papers and most importantly the tools themselves, they should be easy to use and properly documented. And as of next year, 2023, we aim for twice as many tools articles as before. That is, we strive to have 50. And another change is that this time we are going to also include papers that describe experimental methodology or protocols. Again, the idea is to present things that are useful to as many as possible other scientists in protein science. One of my main missions as an editor, at least the way I see it, is to make articles really freely available to read and download for all. Turned out that the Wiley gold number is that we have to double the number of submissions to proteins, double the number of papers that are published in protein science, and of course we don't want to accept poor papers, but that means that we need to find enough good papers to double the number of papers that are published. And indeed, the doubling of number of tools articles is our first initiative towards this. So that's all I wanted to say for introduction. And unless there are questions, I'd like to move on to the science. Okay. So in this talk, I'll present Conserve, which is a tool that was developed maybe 20 years ago, and we are starting, and we keep developing it. The idea is to use evolutionary data to raise testable hypotheses about protein function, and more recently, it's also possible to analyze nucleic acid structures and sequence DNA and RNA. So the idea is that evolution gives us a sense of importance. So typically in a protein, and here we see the influenza neuraminidase, this enzyme and we see in green that it binds some drug in the binding site. So typically the binding site or any other functional region will be more conserved than the rest of the protein, and we see this here. So in principle, if we didn't know anything about the structure of the protein or anything about the protein, then just putting in the evolutionary data gives us a flashlight into the important region of the protein. So that's the basic idea. It's been known for years, and people have been using it. So just to define what is functional importance. Functional importance to me is any amino acid that is involved in enzymatic catalysis or mediation of interaction with proteins or ligands. And then also there will be amino acids that are important to maintain the three-dimensional structure of the protein. So those are also important. Now, the easiest way to look into this, and people have been doing it, is the consensus sequence approach. So look at this example where we have this mini multiple alignment, and in this particular position, we see 6 taxane, and 4 of them are tryptophans. So we say that tryptophan appear in 4 of 6 sequences, so probably it means that it's important in this position. But then maybe all of these 4 come out from primates, so maybe we are better off deleting 3 of them and leaving only 1 of them, and then this tryptophan is no longer important. This position is actually variable because we see that in mouse or tuna fish, it changes a lot. So in general, this problem is called uneven sampling in sequence space, and it leads to the fact that positions can be conserved because of importance for structure or function, and these are the position that we are interested at, or short evolutionary time. And phylogenetic reconstruction may be used to distinguish between these 2 cases. In Conserve, we take advantage of the phylogenetic analysis explicitly, and that makes Conserve more accurate and sensitive than typical other methods. So we use accurate reconstruction of the phylogenetic trees, and we take into account branch lengths explicitly. And I'll demonstrate why branch lengths are important in this slide. So I don't know if you see my pointer, but anyway, in the image on the right and in the image on the left, you see two phylogenetic trees that are identical in their topology and branch length. And in a particular position, in the right tree, we see in taxa number 1, we have a T, and in taxa number 4, we have D, and in the node in the middle, there is D. So that means that in this case, the amino acid retain its identity along the short branch, and then it changed along the long branch, branch, which is expected. But then in the tree on the left, it's the other way around. So the amino acid retain its identity along the long branch and then changed in the short branch. And only if we take into account the branch length, we can account for the different scenarios. So what we do in Conserve is we calculate the evolutionary rate. That is, number of amino acid substitutions per site, per some unit of time, let's say a year. And the link between conserved site, which would be the functionally important site usually, and the evolutionary rate is that a conserved site evolves slowly, and then variable site evolve very fast. So this is the way we estimate evolutionary conservation. In Conserve, there is a Bayesian approach and the maximum likelihood approach. This is for the maximum likelihood approach. For the interest of time, I'll skip it, but the idea is to look for the rate that at a site R that maximizes the likelihood over the data. And the data being the multiple alignment that we built. This is a flowchart of Conserve, and the beginning can be a three-dimensional structure or just a sequence of nucleic acid or amino acid, and then that is Conserve automatically finds sequence homologs, which we can manually select subsets of and use only some of them, or just go on with all of them. Conserve remove redundant and unrelated sequences. It then aligns them automatically, reconstruct phylogenetic tree, calculate conservation using rate for site, and then project the scores on the protein or nucleic acid structure or sequence. In the case of RNA, it can predict two-dimensional structure, and for protein sequence, if it finds a homolog with known structure, it can also predict three-dimensional structure. For users who feel that they know better, then there's a way, and it's easy to do better. There's a way to provide your own multiple alignment and your own phylogenetic tree to get better results, or result that fits better the question that you're addressing. Very soon we will release a new version of Conserve, which is computationally identical, but it looks nicer and more easy to use, and that's what the page will look like. There's an option to use existing structure or a model. You need to specify the chain, and then if you don't know the structure, it's possible to use the sequence. And if it's a protein sequence, then Conserve will use modeler if you can provide the key to modeler, and then it will model the structure. There is default, so you can just push continue and just run with default. But you can change option. For example, you can change the way homologs are searched, and you can change the database to be searched, and the kind of homologs to analyze, etc. So I'm skipping this, but one nice feature here in current Conserve, you submit the job and then you wait and you don't know which stage you are at. But here there is running progress that tells you that now the sequence was extracted from PDB and sequence homologs were found, etc. So you know when to expect roughly the end of the computation. And when it's done, and typically it takes between several minutes to half an hour, then you get the result, and the result will be both a three-dimensional structure of the protein with conservation scores projected on it, and the same thing with the sequence. If you want more data, then there are other possibilities. The coolest thing, which is new, is that now within a click of a button, you get a PyMol or Chimera session with the conservation grades. So you can look at it from different angles and produce nice pictures. But there's various other output that can be useful. I'll skip this slide and just show this example. And again, this is a known case. It's a glycokinase, and we know where the binding site is, where the catalytic site is, and you see that had we not known that, we could look at the most highly conserved region here, and it will show that this is an interesting region. It wouldn't give us a clue of what it is that it does, but it clearly shows that this is the interesting region. And if we zoom in, we see that among the most highly conserved residues are the two known catalytic residues, in this case D205 and K169. That's what the Conserve calculation look with RNA. So if we give RNA sequence, we also get this two-dimensional structure. If we start with a three-dimensional structure, that's the kind of structure that we get. And again, important regions like the anti-codon loop, etc., are conserved as they should. This is a nice feature that I'm not sure that many people are familiar with, and this is, let's say that certain members of a protein family share a specific function. Then you can look at the phylogenetic tree using this tool, Wasabi it's called, that we embedded in Conserve, and you can highlight a certain branch, a certain group of proteins within the family that perhaps share a specific function, and then repeat the calculation using this group, and then maybe get a function that is shared only among this branch rather than the entire phylogenetic tree. In general, Conserve can be used within various contexts of biology, structural biology, membrane biology, genetics, and so forth. I'll show several examples where we use it. One interesting case is, and it's very relevant when we have model structures, and you know that now with AlphaFold we have a lot of highly reliable model structures. Now, typically, residues that are important to maintain the protein architecture are buried in the protein core, and to demonstrate this, I'm just presenting the conserved residues from the most conserved until less conserved and until the very highly variable. And you see indeed that typically the protein core is highly conserved and the periphery is variable. Now, if our model protein does not show that, or from some reason, for example, it shows like a random type of evolutionary conservation pattern, then maybe this model is wrong. So it's a quick way to estimate the accuracy of models. We actually even have an automatic tool that does it computationally, but just visually looking at this, like I showed, is very informative, we see. A note of caution, sometimes, I mean, it's always true that highly conserved amino acids and highly conserved regions are important, but the inverse is not always true. So in this case, this is really the other way around. This is an MHC molecule, and there's a whole genetic mechanism to make sure that it's hypervariable in order to recognize a lot of foreign peptides. So in this case, if we would have been looking for the conserved region, then the most functional important region of the molecule would escape our attention. So, but that's typical of biology. You have to know what you're looking for. So indeed, in Conserve, the peptide binding site is the peptide binding cleft is highly conserved, but is highly variable. But we need to know that this is what we are looking for. And of course, just like in this case, it's highly variable, and in most cases, the functional region is conserved. It can be conserved to a medium degree and so forth. Again, we just need to know what to look for. But because a lot of protein structures are available in the PDB, and because people care about them, we also developed a Conserve DB. That is a set of pre-calculated conservation profiles for the entire PDB, or at least for all the PDB proteins where we felt we could collect a decent amount of homologs to make the computation meaningful. So this is this database, and it saves the trouble of computing. You can just look at it and pick your protein of interest, or do systematic analysis of families of proteins, or proteins, groups of protein that you care about for some reason or another. Because AlphaFold models now are available for many proteins, we felt that it makes sense to also do evolutionary conservation profiles for those, and we are gearing up toward that. We are starting mostly Barak Yariv in my lab, and soon, just like we have the Conserve DB, there will be AlphaFold DB. We will start with human proteins, and we will proceed with other proteins. And just to conclude my talk, in the remaining time, I'll present a case study that deals with cellular signaling through GPCRs. So just to make sure that we are all on the same page, so GPCR are a vectorial way to signal across the membrane. So it starts with a hormone, neurotransmitter, or ligand that binds in the extracellular side of the membrane. And that induced change in the structure of the G protein coupled receptor, GPCR, which changes its interaction with its G protein, and with that it activates the G protein and induces various biological responses. So we noticed that many GPCRs form homo or heterodimers, and very often these are also functionally important, for example, for regulation or other roles. So we were wondering, if there are dimers, then maybe some proteins have the two GPCR domains within the same chain. So we went ahead and looked for them. For that, we constructed hammer profiles for all 90 or so GPCR domains in the ECODE database of domains. And then we searched UniRef 90 for proteins with non-overlapping multiple GPCR domains. And we clustered them using CD hit, and we selected clusters with more than 5 hits to make sure that this is not some kind of noisy data sequencing mistake or whatever. And then we mapped them to representative genomes. And I'm sharing preliminary results. So the first result that we notice is that many proteins have multiple GPCR domains. This is just a selection, and what you see here is this is the sequence the amino acid sequence on the x-axis, and on the y-axis is the number of ECOD profiles, ECOD hammer profiles that include this amino acid. So you can see here this window of around 300 amino acid, which would be a typical GPCR domain, that appears in between 30 and 50 different profiles. So that's definitely a GPCR domain. And here we see another one, and another one, and another one. So in this case, 4. And as I said, we see many cases. You can see here another case with 4 domains, another with 3. This one has 3 consecutive GPCR domains. Most of them have 2 consecutive GPCR domains, but we do see more than that even. And this is distribution of GPCR domains in species. So we can see them in fish through mammals, none in human, but we see them in New World monkeys, and a lot of them are in rodents. Most of them are 2 GPCR domains, but there are even with more. And the most commonly found exemplar, which has 2 GPCR domains, is found in 73 different species. So, because there is AlphaFold, we decided to model it. And we modeled all our candidates, maybe 50 of them. And these are candidates where we are pretty sure that they do share more than 1 consecutive GPCR domain. And this is an AlphaFold model where the level of confidence in the structure is color coded, so that blue is highly confident, and yellow through red is lower confidence. And we can see how the 2 GPCR domains, which are here colored in the center figure, it's colored in green and this brownish thing. We can see that there is high confidence in the domains, but less confidence in how they are packed against each other. And then there is a linker between them, which is highlighted here in red, and there is much less confidence in this linker. This is a 2-dimensional plot of the confidence by AlphaFold of the distance between each amino acid. And you can see that's the image on the right, and you can see 2 roughly squares, each of them, one of them on the upper left and one of them on the lower right, and each of them correspond to a GPCR domain, but the link between the domain, in yellow here, you can see that AlphaFold is not sure about the relative orientation of the domains. We supplement it with, we complement it with Conserve Evolutionary Conservation, as I demonstrated before, and you can see that indeed for each domain the core is highly conserved and the periphery is variable as it should, but then the linker between them, you can see that it's very variable, highly variable, so presumably not important and not conserved enough to cross the membrane. Interestingly enough, in GPCR there are motifs like the dry motif or this NPXXY motif that are linked to their activation, and these motifs appear in both domains. So that suggests that both domains are active as GPCRs. But for that, they have to have the right membrane topology. That is, they have to face towards the outside the part that binds the neuron or the hormone or so, and the inner part has to bind the G protein. So the topology has to be retained. That means that the 2 domains have to be inserted this way. I'm going back to the previous slide that shows that the linker here between them, you can see it in the image on the left and also in the center. It's not structured, so it has a lot of unsatisfied hydrogen bonds within the backbone, and also, and this picture doesn't show it, it doesn't have a long enough hydrophobic segment to form a helix that will cross the membrane. And that's typical for essentially all our candidates. So in order to further study it, we looked into the positive inside rule. And here we color coded in blue all the lysine and arginine. And you can see that this orientation should be the right one because there are more lysine and arginine in the cytoplasmic side than in the outer side, the periplasmic side. But then for both domains to be in this orientation, the linker between them has to somehow cross the membrane, and I don't know how it can do it. So what could be done, and the next slide are pure speculation, is that maybe the first domain enters normally, there's no problem with that, but then the second one, in order to retain the proper topology, it cannot do it because of this linker that is not hydrophobic enough. And here you can see hydrophobicity, and you can see there is no hydrophobic segment which is long enough to form a helix that crosses the membrane. We need maybe 20 amino acids for that, and there's just not enough of those. So here are two possible suggestions. One of them is that there is cleavage in this linker, and then instead of having two separate domains, post-translationally, maybe it's cleaved, and then the other domain enters the membrane, and it will be in the right orientation. Alternatively, and that's maybe more speculative and more interesting, is that the other domain, it gets inserted in some internal organelle, and with that we get a possibility to signal from the outer cell, from the outer environment towards the inner part of this internal organelle. There are evidence that GPCRs can reside in organelles, and there are evidence that they can find their G proteins there. Of course, the ligand is would probably be missing here, but it's a way to communicate. Otherwise, it's possible that the GPCR reside in an upside down orientation in the membrane, where one domain has its N-terminal pointing out and the other N-terminal pointing in, and with that, only one of them would function at any given time. But then because there are two of them, then it doesn't matter if it resides in the membrane in either topologies, it will be fine. Signaling will take place, but only one GPCR domain will be active at any given time. And with that, I'd like to summarize and raise also questions. So there are many putative proteins with multiple GPCR domains, and we see them from fish to mammals up to New World monkeys. Although these monkeys, maybe three of them, they look to me more like rodents. They're like small and they resemble rodents. They are annotated mostly as olfactory receptors, and some of them are chemokine receptors. None of them prevail, so presumably they are not indispensable, but maybe they provide specific advantages under certain circumstances. We are still left with a question of what is their unique contribution, and what is their membrane topology, and what are their mechanisms. And then, since we saw multi-domain GPCRs, maybe there are multi-domains of other membrane proteins. I should say that we discovered multi-domains beta barrels, outer membrane beta barrels. These are the people who contributed to the multi-GPCR project: Ron Solan, Kilik, and Amit, and it was in collaboration with Rachel Kolodny from Haifa University. And I'd be happy to answer questions.
So Nira, I have a question about the linker between the two domains of the two-domain GPCR. So you said that this is found in 73 different species. You may have said this, how highly conserved is the linker, the sequence of the linker?
It's very variable.
Okay.
I can show the slide again. Maybe it doesn't show very well here, but do you see my pointer?
Yeah, it's blue. Yeah.
Exactly. So that's another suggestion that it doesn't cross the membrane.
What kind of amino acid composition does it have, though? Is it just sort of random? Or?
Relatively polar.
I see.
So it's polar, it's not conserved, and I just don't see how it forms a helix that spans the membrane.
And you don't think it could like sandwich in between the subunits and get tucked away that way?
I'm sorry, can you repeat?
Well, is there any chance it could like get inserted between the subunit interface or the domain interface and get like tucked away?
I doubt, because there's not enough space, and also I don't see, if that would be the case, then I would anticipate having a highly conserved region between the subunits, and then also highly conserved region in the linker itself, and I see none. So I doubt that this is the case.
Thank you.
I just think that either they are embedded in the membrane in an upside down topology, and then there's no problem. This linker is just floating somewhere either outside or inside, but then only one of the GPCRs function properly, maybe this one. And then sometimes it will be inserted the other way around, and the other one will be functional. Or as I said, it's cleaved and then no problem, or in some internal organelle, and maybe there's yet a fourth explanation that I couldn't think of.
I see there are some questions in the question and answer. So this is from Alicio, it's not showing me the rest of the name, I'm sorry. Earlier this month, I ran a Conserve job, but used the phylogenetic information and MSA from Clustal Omega just because I was more familiar with it and was running 250 peptide sequences. Question 1, is there a difference if I use MSA from Clustal Omega versus Conserve in terms of conservation values?
Yeah, the results are very, very sensitive to the kind of multiple alignment that you put in. So, for example, if Clustal Omega aligns the homologs differently than by default, we use a program called MAFT. And so if Clustal Omega aligns the homologs differently than MAFT, that would cause difference. But most importantly, it's which homologs you put in. For example, if the Clustal Omega homologs happen to be very close to each other, then and closer than what Conserve provides by default, then the computations will be very, very different, because the analysis was restricted to presumably a very subfamily of the entire relevant family.
And Alicio had a second question. Do we need to use only a single subunit of a heterodimer and trimer, for example, when running Conserve? And he goes on to say, Conserve did not seem to be able to isolate my protein from the complex that was deposited in the PDB search box, even when I typed in the PDB and chain. I was looking at a ribosomal protein, so maybe the initial file was just too much info to search for a single change.
Right. So first of all, I believe that a part of the problem is that is with the format. So the ribosome, as far as I recall, it's in this MMC format that so far Conserve cannot handle, but the new Conserve will be able to handle. So we should be good. And then in terms of the analysis, Conserve analyze one chain at a time. It cannot process more than one. So you have to submit more than one. I'm very hopeful that we will release the new version of Conserve soon, and some of those problems will be resolved.
Thank you. And Paul Whitford had a question.
Chuck, Nir, just to intervene, we have 3 more speakers and we're at 40 minutes now. So I'll let you make the decision, but you may want to take questions.
So then we stop and move to the next speaker. I believe that this is Patricia Clark, right? So yeah, please, Patricia, go on. Thank you, John.
Thanks, Nara. Can everyone see? Can you guys see my slides okay?
Yes.
All right. Good. So thank you very much. Very happy to participate in this session on tools. Tools are incredibly valuable. I'm assuming that most audience members who are listening to this talk are protein chemists and biochemists and from related fields like cell biology, who are interested in producing the sort of biochemical, biophysical quantities of a particular protein. And for a lot of people, that means they're trying to express, for example, a human gene in E. coli or another very common expression host to try to get those biophysical quantities for structural studies or functional studies and so forth. And so the talk I'm going to give today is about a very simple algorithm that we developed called charming, that's for harmonizing synonymous codon usage from one organism to another organism, specifically with the idea of trying to enhance as much as possible, to replicate codon usage patterns, and in doing so, hopefully to enhance functional protein production as much as possible. And so this algorithm is available both as a web server at this web address here, www.codons.org, and also as a downloadable Python script from the GitHub address that's shown over here on the right. Okay, so there's 6 main points that I want to make today about harmonizing synonymous codon usage. And I'm going to first basically explain a little bit of the underlying basis for this and why you may want to think about harmonizing synonymous codon usage, particularly when you're doing something like heterologous protein expression. So first of all, the first point I'd like to make is that synonymous codons are not used interchangeably, and that in particular in coding sequences, they appear in distinct patterns. So just as a baseline thing, I think we're all familiar that the genetic code is degenerate. So for example, for the little 6-mer peptide I'm showing you here, each of the amino acids, with the exception of methionine and tryptophan, each of the other 18 amino acids can be encoded by multiple codons. Those synonymous codons, however, are not used with equal frequency. So for example, in E. coli, the codons that are shown here along the top are the most common, and I'm showing you at the bottom their usage frequencies per 1,000 total codons, whereas these other codons are used much less frequently. But these are absolute comparisons. These numbers I'm giving you here are absolute comparisons per all other amino acid sequence encoding codons. And as protein biochemists and biophysicists, we really take the protein sequence as an absolute. So we take as an inviolate thing that we want to maintain the amino acid sequence. So what we're really interested in is the relative codon usage. So for a given amino acid sequence, are the codons that we're using to encode that protein the most common codons, the rarest codons, some fluctuating pattern of codon usage? What is it? So to address this, many years ago, my lab developed an algorithm, which is also at that codons.org website, that's called %min_max. And %min_max analyzes precisely what I just said, the relative codon usage along protein coding sequences. So this is the algorithm shown schematically. Basically, for any given amino acid sequence, we compare the actual codons that encode that protein sequence to theoretical sequences using the most common codons, the rarest possible codons, or the average codon usage frequencies. And then from that, usually over a sliding window. I'm using a sliding window of 5 here for this example, but it can be any number for a sliding window. We basically compare that how close the actual codon sequence is to those theoretical either maximum, most common codons, or rarest possible codons, or the average, which is 0. And so what you can see is if you apply this algorithm along a coding sequence, so I'm showing you here just for one of our favorite proteins, P22 tail spike, you can see that instead of the rare codons being sort of randomly distributed, they appear in distinct clusters, which also tends to cluster together the more common codons. And I'm showing you here as a reference 200 basically randomly generated synonymous versions of tail spike, just to show how distinct these dramatic patterns of rare and common can be. So this is basically synonymous codon usage, a particular synonymous codon usage. And how that synonymous codon usage, one of the reasons why you may want to think about synonymous codon usage is that it can affect aspects of protein, functional protein production. One example, which my own lab is very interested in, is that synonymous codon usage might affect co-translational protein folding by the following mechanism. So for example, if I'm encoding a protein with every so often a group of rare codons, the rare codons in general tend to be translated by the ribosome more slowly than common codons. And so those rare codons, once the ribosome encounters that portion of the mRNA sequence, that would give the portion of the protein that had already been synthesized an opportunity to get closer to an equilibrium conformation before translation continued on. Versus if I encoded that coding sequence with only very common codons, I may be able per unit time to run off more copies of that protein, but the folding yield might be much lower. And so people who have tried heterologous gene expression in different organisms often see that what happens is that the patterns of rare and common codons can change. And a long-standing strategy in the field is just to try to get rid of rare codons and make very common codons. But again, one of the possible outcomes that happens occasionally is making a whole lot of protein, but that protein is not very soluble. So my own lab, as I mentioned, is very interested in these effects of synonymous codon substitutions on protein folding. And I'm going to give you 3 short examples here of how that can work in experimental examples before I describe the codon harmonization approach in charming. So many years ago, we developed a protein to try to explore this idea of the extent to which synonymous codon usage can affect protein folding. And that protein we call YKB. And YKB consists of 3 half domains made from fluorescent proteins. And so depending on whether basically the first and the third half domain are competing with each other to interact with the central half domain. And depending on who wins that competition, we get either a yellow fluorescent protein or a blue fluorescent protein. And our hypothesis was that because the yellow half domain is synthesized first, that we would expect if this protein is folding co-translationally, that we would get more of the yellow outcome. And that's indeed what we see. Furthermore, we hypothesized that if we encoded the 5' end of the B half domain with increasingly rare codons, we would give Y even more time to interact co-translationally with K before B appeared, and even further skew that yellow to blue protein outcome. And that's precisely what we see. So we changed the codon rarity using this percent min max algorithm for only an 18 codon window at the 5' end of B. And we were able to titrate in this YK to KB fluorescent ratio. So this was a really pivotal result for us because it showed that it was really the first demonstration that protein folding and protein structure can be predictably tuned by changing synonymous codon usage in the cell in a predictable way. It doesn't, however, indicate that nature is using synonymous codon usage as a way to tune protein folding. And as a first approach to that, we undertook a very large multiple sequence alignment based computational approach to see whether rare codon clusters like the ones I've shown you previously occur at conserved positions in the multiple sequence alignment. And our rationale here was that because homologous proteins tend to have very similar structures, that if rare codons, as one example of a pattern, are important for co-translational protein folding, then we would expect them to appear at similar positions in that multiple sequence alignment. And that's what we saw. So 18% of the homologous gene families that we analyzed, we saw conserve patterns of rare versus common codon usage, one of which I'm showing you here. So the green bars in the background indicate statistically significant enrichment of rare codons in this central region of the single domain protein. And more recently, we've been analyzing the effects of synonymous codons on organismal fitness, particularly for this E. coli antibiotic resistance protein called chloramphenicol acetyltransferase, or CAT. So CAT, like all coding sequences, has a non-random distribution of rare and common codons. And we made an alternative sequence shown here in red that had the same overall fraction of rare and common codons, but different local patterns. And what we saw was when we expressed the sequence in E. coli, that we had a dramatic twofold decrease in growth rate for E. coli, expressing these proteins, either the wild type or the synonymous version, in the presence of the antibiotic. So in this case, we were able to trace back part of the effects of these codon substitutions on a change in the extent to which the resulting protein is prone to degradation by a AAA plus ATPase, ClipXP in vitro. So I hope those are convincing evidence that synonymous codons can change and affect co-translational protein folding. I'll say they can also affect many other things. And so, for example, we spend a lot of time in my lab, because we're interested in studying effects on folding, we spend a lot of time controlling for and normalizing something that we see a lot of, which is effects of synonymous codon substitutions on protein level. So I'm showing that here for the two proteins I just showed you, wild type and the synonymous version of CAT, chloramphenicol acetyltransferase. And so the codon harmonization program I'm going to talk about with you today was really started when Annabelle Rodriguez, a grad student in the lab at the time, asked the question, well, if I start with my SHUF1, my synonymous sequence here, and I alter the codon usage to recapitulate the patterns of codon usage in the wild type sequence, will I recapitulate wild type protein production? And so that brings me to my next point, which is, in fact, as I'll show you in a moment, recapitulating wild type codon usage patterns, and really many different patterns, not just synonymous, not just relative codon usage frequency, which I've showed you so far, can be an effective tool to recover wild type protein accumulation. And the other related point I'd like to make is that synonymous sequence space is astronomically vast, and that presents both challenges and opportunities for designing these synonymous sequences. So far I've been telling you about this percent min max algorithm that my own lab identified to try to think about how synonymous codon usage might affect co-translational protein folding, given the known association between rare versus common codons with the local elongation rate of the ribosome. But I'll point out that there's many other metrics or measures that models that people have developed to measure what's optimal for codon usage. And so the oldest of these is the Codon Adaptive Index, which compares the codon usage in a gene of interest to the codons used in the most highly expressed genes in an organism. And there's many, many others here as well. So we're agnostic. We develop percent min max, but we don't really care which model is the best fit for protein production. We just want to figure out what the best one is. So ideally, what Annabelle wanted to do was start with our SHUF1 sequence that had reduced protein accumulation and harmonize that SHUF1 sequence back to the wild type sequence using not just the percent min max profile that I'm showing you here, but several of these profiles. So that would yield a sequence like the one I'm showing you here. Whoops. In brown, that's Annabelle's harmonized sequence that now matches much more closely to the wild type codon usage, but uses different codons to achieve that. So we're harmonizing to the pattern using different codons. Okay, so the second point I wanted to bring up is that this idea that synonymous codon space is astronomically vast. So even just for the little hexapeptide I'm showing you here, we have many hundreds of possible sequences that can encode that amino acid sequence. And this is just a little hexapeptide toy example, given that the average coding sequence, even in E. coli, is around 300 amino acids, and there's on average 3 codons per amino acid. This means that even for an average sized protein, we've got more possible synonymous sequences than we have particles in the universe. So this is all to say that there's no way that we can exhaustively sample synonymous sequence space. We have to sample sparsely. But that provides an opportunity because it means that, it means that we should be able to design sequences that correlate strongly with one codon usage model, but not others, and really be able to distinguish, is one model more predictive of functional protein production than others? And this is a longstanding question in the field. So this is what Annabelle did. She used this algorithm Charming, which I'll explain in detail in a moment, to design synonymous sequences for CAT, the protein I've been talking about. So these are shown here. So again, in black, in both cases, is the wild type sequence. In red is the sequence that Annabelle made that's harmonized closely using this Charming algorithm to the wild type % min max codon model. And in blue is a sequence that she harmonized not to % min max, but to this alternative model called TAI, which takes as optimal to what extent a codon correlates with the tRNA abundances in a coding sequence, in a genome, in an organism. So you can see that Annabelle was able to, because of the vastness of synonymous sequence space, to select harmonized sequences that match well to one profile, but not to others. And she generated in total eight different sequences, 6 which, like the 2 I'm showing here, were harmonized to different codon usage measures, and 2 others that were harmonized that were not harmonized, that were actually just also synonymous sequences, but using other ideas. So one is the conventional codon optimization procedure. So this is using a GeneArt, Thermo Fisher's GeneArt algorithm, which is a proprietary algorithm, so we don't know exactly how it works. But you can see that if we analyze the output from GeneArt using our % min max metric, that it's mostly encoding the sequence using very common codons. And another sequence that Annabelle made was this scrambled sequence. This is taking only the codons that are used in the wild type sequence and redistributing them randomly in the sequence. But again, keeping a synonymous sequence. So we're always, in every case, still producing the wild type protein sequence, just using different codon order in the case of this purple sequence. So although each of these sequences only has 40 to 50% codon identity with all the other sequences, each of the other sequences, we were able to select sequences that all have very similar predicted mRNA stability and predicted or known GC content, right? So we're able to keep some variables constant and vary only the selected variable. So the bottom line here is when Annabelle expressed these harmonized and the not harmonized sequences in E. coli, what she saw was that in general, harmonizing works. And it really doesn't matter which metric we use. So for the 6 harmonized sequences, which are shown here in gray, 5 out of the 6 recapitulated the wild type protein accumulation. And more so than the non-harmonized sequences. And this includes that wild type scramble sequence I showed you, which has the only difference between the wild type coding sequences is just changing the codon order. Okay, there are some glaring exceptions here. One is the sequence that we harmonized to CAI, and one is the sequence that we harmonized to that we optimized using GeneArt. So they produce very, very little protein. Now, we spent a lot of time figuring out why this is. And what I would suggest for the average run-of-the-mill user is that you not spend a whole lot of time trying to figure out what's going on here. If your sole goal is just to produce as much as protein as you can, just go ahead and make some other sequences similar to our approach here. Because I'm not going to get into this today, but what we found out after a lot of trouble is that this GeneArt and CAI sequence have much less protein accumulation, not because of anything to do with co-translational protein folding, but because synonymous codon substitutions can have many other effects on functional protein production. And so again, one of the things about using the metrics and harmonizing to wild type codon usage is we're sort of agnostic to what the actual mechanisms are that are going on. We're just saying we want to recapitulate the wild type codon usage pattern. Okay, so I'm going to talk in detail now about how these algorithms work that I'm talking about. So I'm going to talk about some specific slides about the very first step of the Charming algorithm and then the core Charming algorithm, the workflow. So codon usage frequencies vary significantly from one organism to another. So I'm showing you arginine codon usage in E. coli rank from most common to least commonly used, and then conversely in human. You can see that the rank order is very different. And so what this means is that the most common codon that's used to encode arginine in human is actually one of the least frequently used in E. coli. And what that leads to is a mismatch between these usage frequencies can lead to very different profiles. So if you focus here where this arrow is, you can see that because this sequence has one of these very commonly used arginine codons in human, when we instead translate this protein in E. coli, that's a very rare codon cluster. However, if we just re-rank the codons in terms of the order, the frequency order here in E. coli, so if I am using the most common codon in human, then I want to encode that with the most common arginine codon in E. coli, you can see that that ranking itself, as shown by the green line on the right, is already able to largely recapitulate the wild type codon usage pattern in the host organism in E. coli now. It's not perfect though. So with the charming algorithm, so this is a first step of the charming algorithm that we call Rodriguez initialization, because it was developed by the student Annabelle that I mentioned. The charming algorithm goes further, and you can see that there's still significant discrepancies between the rank order initialized sequence and the target sequence, the black sequence. So the charming algorithm basically further enhances the harmonization with the wild type codon usage in the host organism. So I'm running short on time. So the steps of the algorithm, this is a figure from the paper. So I'll definitely urge you to look at the paper if I don't have time to go through this in detail. But basically what the algorithm is doing is it's taking in the original organism, the wild type codon sequence, calculating those target model values, again, using CAI or percent min max or any other target model. Then the deviation between that wild type original organism profile and the profile in the expression host, that deviation is calculated. So for example, this codon position is labeled in orange because it deviates significantly higher above the wild, or just higher in general, doesn't have to be significant, higher than the wild type codon usage in the host organism. And so we identify CDRs, which are regions of 10 or more codons that deviate in one direction relative to the wild type codon usage. We identify, and the paper describes in detail how these are identified, regions within that CDR to modify. And then we start modifying one at a time the individual codons within that CDR to try to match more closely the harmonized or the target sequence. And this process iterates until basically there's no additional improvements that we can make in the codon match, meaning the deviation as measured by the Pearson correlation coefficient between the original organism target model values and the generated sequence. Okay, so what I'm showing you here in red, these dots here, this is looking at for just Rodriguez initialization alone that we're able to, actually, I'm going to skip this slide in the interest of time. So just what I'm showing you here in gray is the results for the deviation. Well, this is the deviation reduction overall. So the closer my histogram is to 1, the less deviation there is between the pattern that I'm going for and the one that I have, the sequence that I have generated. So you can see that Rodriguez initialization alone reduces a significant amount of the deviation between the initial starting sequence and our optimum target. Charming alone without initialization also does a better job, and the two together actually do a really effective job at getting very close to the matching perfectly the target sequence. Okay, so these are the points that I covered today. I'm going to end by thanking my lab, and in particular Annabelle Rodriguez from my lab, a recent graduate who is the real champion for this project, and worked closely with Gabriel Wright, a student co-mentored by Scott Emmerich and Tiana Milenkovic at Notre Dame, who was the real brainchild and computer science student behind the charming algorithm. And I'll stop there and take any questions.
So there's time for really one quick question. So I'm reading just one by Moshe Goldsmith. Have you looked for a connection between the use of rare codons and the recruitment of chaperones during protein translation? Could ribosome posing be used for folding of large and complex proteins with or without chaperones?
That's a great question. So one of the hypotheses we explored in the cat study that I mentioned was whether producing this cat protein that folds to a slightly different structure, whether that would trigger induction of the heat shock response. So whether that would lead to more production of chaperones. We didn't see any evidence for that this was using quantitative mass spectrometry, but it's certainly possible. And it's also possible that even just with the constitutive level of chaperones, that additional chaperones could be recruited to those ribosomes. We did not test for that.
Okay, so thank you very much, Patricia. And let's invite Paul Whitford to share his screen and start his talk.
To thank the Protein Society for putting this together and for giving me a chance to tell you what we've been working on. I'm going to tell you about a class of models and software we've been developing called structure-based models, or the SMOG line of software. And I'm going to give you a little bit of background on what these models are. There's a wide range of definitions one could use, and highlight some of the applications of the tools we've been developing, so you have an idea of how you could potentially use these tools in your studies. Oops, it's not. There we go. So what is a structure-based model? There is one feature that makes a model a structure-based model. These are, when we say a model, we're talking about a potential energy function. And we're saying that at least one element of that potential energy is defined based on knowledge of some structure that is out there. So this could be every interaction stabilizes a native fold. It could be that only some terms stabilize a specific configuration. There's really any sort of definition that has any sort of nativeness in the force field makes it a structure-based model. So these can be models that are elastic network models. You could do explicit solvent simulations and put native-like interactions on top of them. As I said, you could have a fully native model, a fully native model. You can pick different types of resolutions. So if you look in the literature, there have been groups that have published models that are coarse-grained at the C-alpha level, C-alpha, C-beta for proteins. RNA and DNA have 3-point models as well as 1-bead models. They're all atom variants, implicit or explicit solvent models, explicit or implicit ion models, electrostatic models. There's just sort of an endless number of variations. And again, the only thing that is common between them is that something is based on a known structure, as little as one interaction. So I like to highlight this because a lot of people have ideas of structure-based models meaning a specific thing, meaning a specific resolution, and that's really not the case. And so what you, as a take home, if you get nothing else, you should always remember no one size fits all for these models. It's really a class of a structure of a type of approach for looking at dynamics, but you really have to think about what is important for your specific application. So you may need high resolution models, you may need coarse grain. You really need to think about what you're trying to ask when you're applying these models. So please take some time and think about that. And if you see someone presenting work with structure-based models and they make big claims about a result, make sure they tell you what the model is and why they chose that version, because there are so many different things one can do. In addition to the variability in the model's definitions, you can also use any sort of classical techniques for studying these models. You can do traditional MD simulations, Monte Carlo methods, normal mode calculations, discrete path sampling, enhanced sampling approaches. All of these can be applied once you've defined your force field. And if you look in the literature, you will also see various names that appear for these models. We call them SMOG models because it's the line of models or the style that we've developed and shared. A lot of the times you'll see them called SBMs or Go-like models as well. However, the term Go-like model typically refers to coarse-grained variants. So a little bit of background of where these models have come from over the last 3 decades. Back in the 90s, these models were lattice-based models where every residue was a bead that moved on a lattice and beads could interact with one another. Around 2000, Clementi came out with the C-alpha model, which became a very widely used model where you could map a model to a specific protein. And then in the mid-2000s, which is when I started working on these models, there were a lot of efforts to extend the models to include various levels of energetic detail, being electrostatics, solvent effects, or, and what I was doing was looking at multi-basin representation. So trying to describe not just dynamics about a single minimum, but dynamics between minima. If you have two competing structures, how do you study those transitions? Then in the mid-2000s, we also developed the all-atom versions of these models. Other groups had developed similar models, but we developed ours around that time as well, and we made them freely available through the web, the SMOG web tool. And then more recently, what I'll be telling you about more today is the SMOG 2 line of software, where we've been trying to, again, expand the possible applications of these models and make them more easily accessible to the broader community. So I will share a little bit about how the technology works and what you can do with it, and also give some highlights from some recent studies, just to give you an idea of how far things have come from those early lattice-based models of proteins. So first, what does our software do? Well, when you have your whatever biophysical question you'd like to address, you have some system that you'd like to study, you need a structure file. Our software, the SMOG software, takes that structure file and then generates the force field. So it defines all of the interactions that will govern the dynamics. We've written this in a way where you can take the output and then use any leading MD package or numerical package that you would like to use. So they're compatible with GROMACS and AMD, OpenMM, the Wales approach for discrete path sampling. There's also some marginal support for LAMPs as well. So we're really working at this intermediate step of defining how you would like to ask your questions. And then you can really turn to the professionals when it comes to performing simulations and use really highly tuned software to get the best performance and use whatever hardware you would like to use. So what does a structure-based model look like? It looks like every other model. As I said, these are completely general definitions. So this would be the potential energy function. You have some terms that maintain the backbone geometry. Then you have some terms that give you dihedral energies, something with related contacts, and something for excluded volume to make sure that atoms don't pass through one another. Now, all of these elements can be adjusted and tuned depending on what you are trying to do, depending on if it's a coarse grain model, all atom model, or if you wanted to have a model that has various forms of dihedrals, various forms of contacts. These are all free parameters, although we do share sort of some standard versions that we use commonly. The user is actually at liberty to make any changes that you like. So when we first started working on these models back in José's group, back in the mid-2000s, Jeff Noel and I were developing these tools, and we thought, it took a lot of effort to get them to work for anything. So maybe people will find it useful if we can all sort of do things in a similar way so that we can start to compare our results. So we don't have 1,000 different variations of models that are fundamentally different in a way where you don't even know if your results are due to differences are due to the proteins you're studying or some intricacies of how you set up your specific model. So we released the SMOG1 web tool, and this is a real simple tool that you just upload a structure file and it will give you all the files you need to run a simulation in GROMACS. It's very rigid, and I have to just admit it was built on Fortran, so this is very archaic code that we're not going to give to anybody. It's just, it runs the web page, and that's why we did not want to distribute it. But it has been used quite a bit over the last 15 years or so. So it is a reliable tool for at least getting your feet wet with structure-based models and starting to play around with different types of systems. Then in around 2015, we released SMOG 2. So SMOG 2 is really a significant step forward in what you can do with structure-based models. So it is, instead of being a web tool, it's a downloadable version of the code. You can download it as a container, source code, or run it through Google Colab if you want to. And really what makes SMOG 2 useful is that it's portable. So you can define your own rules for constructing force fields just by providing XML formatted files. So if you are working on your protein and you think that a certain way of distributing energetics, say using an HP model versus an electrostatic model, is more useful, you can just build your definition for a force field. You publish your results, and then you can tell anyone out there, if you want to use my model, here are the templates, you can apply it to whatever system you would like to study. So it makes a way that the field can actually have reproducible results, which is, of course, very helpful. It's also designed with versatility in mind. So as I said at the beginning, the structure-based models are very broad in definition. And so we've tried to construct this in a way where you can basically add any elements from the amber or charm lines of force fields and put them right on top of your force field as well. So if you want it to include electrostatics or explicit ions or novel ligands modifications, all of these features can be implemented without the user having to do any sort of source code modifications. So you just define your XML files, and then you have a new construction of a structure-based model. We have also extensively tested the software for large and complex systems, so it's a very reliable code. And we provide a lot of tools for additional levels of customization. So as you get into your system, you might say, oh, well, I think this particular few residues are very important. How would I just remove those in a very specific way, or I make them a little bit bigger, a little bit smaller, I add the charge, remove the charge, and you can start to tweak the various features in your molecule and ask what sort of large scale effects might that have on your system. And then also say with SMOG2, we've released a library called OpenSMOG, which works with OpenMM, which just further generalizes the construction of these models. So you can really put in any non-bonded terms, any contact terms that you would like now. And with a single GPU, you get the performance of about 500 to 1,000 CPUs with our models for anything sort of large. So that's the software side. Now I'll spend the rest of the time telling you about some recent applications, so you have an idea of what one can expect if you were to use these tools. What my group has done, the largest focus has been on the ribosome over the last 10 years and looking at large-scale conformational rearrangements in the ribosome. So the ribosome has 150,000 to 200,000 non-hydrogen atoms, and in our models, we include all of them. And we look at conformational changes that are on the scale of 20 angstroms to 100 angstroms. And with these models, you can, while they are still approximate models, you can watch spontaneous barrier crossing events. So we've sort of worked our way through the elongation cycle through a range of studies, and not just simulating a single event, but simulating hundreds or thousands of events, and then going back and saying, well, like I said, if this helix is important, let's remove it. What effect does that have on the dynamics? Let's put in electrostatics. What effect does that have on the dynamics? And over time, you just build up a more sophisticated or more complete picture of your assembly. So it's been working very well for the ribosome over the last 10 years. Very recently, just to give you an idea of what a typical simulation can look like, this would be a yeast ribosome where we were looking at subunit rotation. And so first, what the movie is going to show you are just the endpoints of rotation. The system will rotate so that you can see how the small subunit moves with respect to the large subunit. Let's unrotate it and rotate it. This is about 200,000 atoms that are included in every simulation. And it's about an 8 degree rotation of the small subunit, which itself has about 80,000 atoms. So we set up our structure-based model, we let it go, and eventually we will see these spontaneous large-scale rearrangements occur. So there you see about an 8 degree rotation of this, more than a 1 megadalton subunit. And then it will back rotate, and we just let it keep going, and you can collect statistics on these transitions, and you can do it to such an extent that you can actually begin to calculate free energy barriers and start to identify what are the interactions that are forming at the transition states, which give you direct connections to experiments, because you can start to say, well, these residues are more important for kinetics, these residues are more important for stability, and that can be tested experimentally. But with all of that, I always like to stress these are really still toy models. So we're not expecting to get the full picture, but it's a way of saying, here's a baseline description. And now as we start adding perturbations, just like if you had a car engine that you didn't know what was going on, you just take a screwdriver and turn things, we can just look at these machines, turn pieces, and say, well, if we twist this or we poke that, what effect does it have on the large scale dynamics? And we can test that with these models. So you can really start to see how a system responds to various external stresses that could be applied. Another fun example from this last year was looking at the spike protein from SARS-CoV-2. So everyone here has certainly seen plenty of movies and images of the spike protein. It's all over the media, but you probably haven't seen it undergo its fusion-related conformational change. So what the protein does is it recognizes the host cell, and then it has to recruit the host cell and bring the viral membrane to the host membrane, which allows membrane fusion to occur. So what we were doing with this application was we were specifically looking at how glycans can affect this rearrangement. So in the simulation, the orange are the glycans. What we were showing is that just the excluded volume of the glycans actually impedes the motion of this rearrangement, and it will pause in configuration like what we're seeing right here. And by pausing, it allows these fusion peptides to just reach out further and gives them more time to try to recruit the host cell. So again, while this is a first pass picture of the motion, the role of glycans, the way we modeled them, was just sterics. So excluded volume of the glycans induces the slowdown, and the sterics are going to be present in any real system, and they're going to be present in any more highly detailed model as well. So we'd expect this relationship to be robust. And there is the conformational change as it completes, where basically the protein is flipped inside out and upside down and reordered about half of the helices. So you can now start to actually go in and again start tuning these systems and tweaking them and asking, well, are there hotspots associated with a conformational rearrangement? Is helical propensity important in a certain region versus another region? And really think of it as a way of dissecting the relationship between the different components of a larger assembly. We've also taken some time to try to look at viral capsids. So here are a couple of examples with SMOG2 that we've applied. We looked at the HIV-1 capsid. This is the same model from the Schulten group that they did explicit solvent simulations. In our case, we just took the capsid itself, which has 1,356 proteins and about 2.5 million atoms. And we have a model for ions that we're releasing very soon, where we can actually put in explicit monovalent and divalent ions. So as I said, this is very, very far from what you would typically think of as a structure-based model. This is an electrostatics plus explicit monovalent plus explicit divalent ion model with effective potentials, although there is this element of the structure-based potential underneath the capsid. And in this application, we were able to actually look at how the ions over time condense onto the viral capsid. You could start to ask, how do monovalent versus divalent ions change the local electric fields, and potentially relate that to some sort of functional role of the virus. Another fun example recently was looking at the T7 bacteriophage. And during maturation, the capsid undergoes a large expansion process. And during that expansion process, all 420 N-terminal tails transition from a disordered state on the inside of the capsid to an ordered and interlocked conformation on the external side of the capsid. And with these models, we can actually start to look at this motion as well and start to ask, how do the different positions of the monomers affect the dynamics of these tails? Are there some strong, are there some strong steric features that would at least sort of enforce a specific sequence of rearrangements or not? And we actually did find a very strong signature where specific sites in the hexon were slow, other sites were very fast, even though these are all the same protein. So we can, again, this is about 1 million atoms. You can set it up with SMOG2, run it with Romax, run it with OpenMM, however you want, and start to look at these very large scale rearrangements, and then go back and start adding all of the other details that you might want to account for, maybe in this case, including DNA. Just to highlight the reliability of the code, we've also tried to break it as much as possible. So we made some hypothetical systems just to see how far we could take the tool and whether or not there were some technical limitations. So we set up a hypothetical model of a 1,000 nucleosome system. So this is a crystal structure of a single nucleosome that we just tiled into this long extended region and put some linkers between them. So it has about 184,000 base pairs in the DNA strands and 1,000 nucleosomes or 8,000 histone proteins. And that's about 3 million atoms per DNA chain. And it works. You can run it. You can start playing with these systems as well. We didn't do anything more than simply making sure you could set it up so that people in the field who are interested in looking at chromosome simulations can do so. We also consider crystallographic lattices because in cases like the ribosome, you might want to look at flexibility of a system and how B factors could give you suggestions of the flexibility. Well, in the crystallographic lattice, there are a lot of opportunities for crystallographic effects to perturb that flexibility. So we said this was a nice example of asking to what extent do crystallographic contacts affect the predicted B factors with a model. So this is a 15 ribosome cluster. We set up larger clusters up to about 150 or 160 ribosomes. It all runs just fine. We also took a real classic protein CI2 and tiled 36,000 of them. It also works. You can do those simulations. So we really think we've tried to break the code as much as possible so that basically no matter how wild your ideas are, this infrastructure will work for you. So I want to thank everybody who's contributed to these efforts through the years. I've worked very closely with Jeff Noel and Jose Onuchik in directing and initiating the software development side. We've had many active contributors from my group and Jose's group over the years. And I also want to say thank you to all the users who are not in our groups working on this actively, but who give us feedback, because we can only imagine the sort of creativity that people have. So the more creative you are, you might find a limitation of what our code does. Let us know. We're always happy to try to make it more versatile so that you really have like a one-stop shop for building structure-based models. So this is just a small list of people who through the years have, who either I know who have emailed me suggesting various improvements to make things work better. And with that, I have to thank the NSF who's been supporting this work for the last 15 years. Over the last year, we had a lot of computing resources from AMD through their COVID-19 HPC fund. Research computing staff at Northeastern and Rice University have been instrumental as well, keeping those computers running. We just can't do anything without them. And this has all been done in the Center for Theoretical Biological Physics. If you're interested in performing large-scale simulations, we also have some workshops coming up in the next year. Tentatively titled Simulating Ribosomes, Chromosomes, and Other Large Assemblies. So if you'd like to learn how to do it, come join us. We're happy to tell you. We really want to make these tools accessible to everyone. So with that, I'll thank you for your attention and answer any questions.
Thank you very much, Paul. This is very interesting. I actually wasn't aware of this resource. It's really fantastic. I just have one question. So, and it's a general thing that I was debating over also. So, when you do simulation and the input and in the end you get a motion. So, how can you design experiment to check whether the simulation is anywhere near reality? Can you give a specific example, like maybe with the motion in the ribosome or anything like that?
So I can give an example for the ribosome. Recently, we were looking at a motion called PE formation. It's about a 40 angstrom motion of the tRNA. So we calculated the free energy barrier. We identified where the transition state is, and we said, oh, these interactions are formed. They transiently form. So what we did is we took our model and said, well, it seems like the steric contribution is significant here. So let's just make the atoms a little bit bigger and repeat our calculations. And make them bigger, I mean, half an angstrom larger, just slightly more repulsive. And what you see is a slowdown of about an order of magnitude in the kinetics, because the tRNA just barely can fit through this passageway. And we can then go back and say, well, let's not just perturb all of the residues, let's change just one of them or two of them. And you can really start to hone in on specific residues that seem to have a stronger steric component. And in that example, then one could imagine just putting a more bulky side chain in that location would be sufficient. If our overall picture is okay and sterics is important in that transition, then just making it a longer side chain should be enough that you'll see on the order of, say, an order factor of 10 decrease in kinetics. So you do have to go through an iterative process to connect to experiments. But that's what's nice is even the ribosome, which is fairly large, nowadays we can run on one GPU 10 times faster than we could run on 300 cores 10 years ago. So a one node machine is now equivalent to like 8,000 cores from 10 years ago. So you can actually run hundreds of ribosome simulations in a week on a modest sized cluster, and then have to think about how to analyze it. But then you can go back and say, well, I think this is important. Let's tweak something. Let's remove this helix. Let's move this around, and see if your way of thinking is at all physically rational. And if it is, then you can try to even tune the perturbations to reflect more of an ideal experiment, which you could then interface with them. So there's a lot of avenues, and that's what we've tried to build into this computational infrastructure, is to let people do that.
Okay, thank you very much, and we just finished time, so let's thank Paul, and then Vladek, your turn now, and this will be the last talk for the webinar. Thank you, Paul, again.
Thank you.
Okay, I am sharing my screen. And wait a second. Okay, do you see my slides correctly?
Yes, we do.
Yes.
Okay. This is the title of the paper, which was published in Protein Science Tools, and but really thinking about this paper and about this talk, I am going to talk how to get the best optimal structure from quite often suboptimal data, suboptimal diffraction data. And first slide is disclosure. I'm involved in various commercializations efforts, and this is the real problem. But the fraction experiment is the last experiment before the position to PDB. Many people claim that data set can be collected in 2 minutes. If this would be true, our capacity now, looking at the synchrotron station, is we could produce 350 times more than people produce, which people deposit to the PDB. So our efficiency is 0.3%, not very good. Why is that? Because data reduction may take 5 minutes, structure determination between 5 minutes and infinity, and structure refinement, it depends. But it can be done very fast if data are collected well. And this is the problem. So if we will look at number of structures versus time produced by 24 most productive in sense of number of structures synchrotrons in the world, there is quite interesting picture, and I would like to look for details into nuclear instruments and methods paper. But what we put also overall structure quality, median of overall structure quality, and it's also published how this was calculated. And you see that the quality for various synchrotron varies. Yes, that it's not the same. And we may ask, okay, the synchrotron station has many limitations. It's X-ray flux, detector, computer speed, experimental protocol, software, coordination of software at the beamline with software which is done outside. But what is really critical is integration of all of that. So many people are convinced if they replace their 1 million detector for new 2 million detectors, which is much faster, everything will change. No, the answer is no. Because we will collect data very fast, but so what? Our goal is not to collect as many data sets as possible, but our goal is to determine as many important structures as possible, but these structures have to be very good structures. Yes, and the question is, what does it mean, very good structure? So where are the real bottlenecks? This is the HKL 3000, very simple show of various steps. At the end, we have the structures secondary. Yes, but it was published. I didn't put all papers. It was published in many papers. It was started in 90. Program was started in 90, and now has around 40,000 citations. So it's one of the most cited paper ever. But really, the problem is not the problem. The problem is sample. And we have to realize that protein crystal is much more expensive than any diamond. If you will calculate, if you will take into account all effort and money which is going to produce good proteins, protein crystals. The same is with cryo-EM. We have to produce grit, and it's not easy, as people think. Yes, we do not need to produce crystals, but we still need to produce grit. So this is the real problem, and how to make distinction between good crystal and poor crystal. Data collection. Last experiment. And the question is, what is beneficial for particular type of experiment? And some beamlines have personnel who shows people what to do. Some people are not educated in the lab. There are less and less X-ray system in the lab. And there are two things: what is possible and what is beneficial. So if our goal is molecular replacement, we should collect low resolution data. We have to have low resolution data. If we are doing SAT or MAT, I would say medium, but very accurate differences between anomalous data. Ligand screening, very high resolution, and the same for final refinement. So, and we have very few choices, in fact: wide sector versus narrow sector, inverse beam experiment, crystal orientation. And we were very successful because quite often we were doing things against the common sense, or not common sense, but common beliefs. Yes, they're not always common beliefs. So what is critical is a site file. What is site file is the description of the experiment system. But it's not only description of the detector, but a description of the goniostat, how the goniostat moves. You have to have all that information. And HKL have so-called site configuration, which you can put things by hand, or you can process lysozyme and by one click move the data from the processing of the lysozyme into site file, and you have perfect site file. Quite often, people are saying, oh, with site file, only X beam and Y beam is important. And this is in some sense true, that if you have wrong position of the beam, you are getting misindexing. Yes, and it's difficult to go out from the misindexing. And if you are looking in indexing, the Brave lattices, which you are here, only perfect is for primitive triclinic, but in reality, this is primitive orthorhombic. So you see that this is the result if your position of the beam is wrong. But why position of the beam is wrong? How do you like this image? Not very much. Yes. This is the same image. But the only difference is exposure time. And now what people quite often are doing, they are saying, okay, we have great detectors. They have zero noise. It's not true. It's zero readout noise. There are still noise. And you see in the background, you have zero, but you do not have statistics. So you're really looking at the image, you do not know what is going on. Yes, you do not know if this is even a crystal. And you have to sum crystals, to sum diffraction images, narrow diffraction images, to see what is going on. And sometimes, for short exposure, you do not see that you really do not have one crystal in the beam, that you have here 3, 4, and I have seen 5 or 6. Now, if it's the project of the class of ribosome, yes, you can process this data. But if it's standard project, you should go, I mean, by standard may be very important, but you should really go back to flash cryocooling. Yes. It's much, much faster to do that. To think a moment how I should do cryocooling of my sample. Now, this, you may think that's a detail, but you are getting suggestions of Brave lattice. I would like to say that it's only suggestion. It does not mean, and people are saying, oh, HKL shown me that it's hexagonal lattice. No, it's suggested that it's hexagonal lattice. It may be not. And there are additional tools which allow you to check really if the space group is correct. I can tell you one thing, that in PDB, there are thousands of deposits which are done in wrong space group. And in that case, the structure may be correct, more or less correct, but not very optimal. Now, we made, we got grant, big data grant, and we made proteindifraction.org, which has various people may deposit raw diffraction data. What we have learned, the data collection strategy is taken from the sky. Yes, you should have some dependence between unit cell size and how large is the sector. Yes, because you have overlap of reflections if your sector is too wide. But as you see, people like 1 degree, people like ½ degree, people like 0.25, but it has nothing to do with the units. Yes, it's just the number which I like. The positions of the beam should be in the header. You see that this is over time and difference between the real position and the position which is in the header. It has nothing to do with the reality. It's just the number. And there is assumption that people will figure out. If everything is correct, we can use automatic correction. And automatic correction are taken into account that you have one reflection measured many times. So you can try to take into account absorption, radiation damage, and error model, better error model than you have. And what else? Well, very, very important, that you can take into account an isotropicity of the diffraction. Diffraction is not always isotropic. The problem is that if you are doing that, sometimes reviewers complain that that does not follow the standards which were done 30 years ago, and 30 years ago, nobody was thinking to take these things into account. Data statistics before and after correction, you have everything is much more smooth, and this is obvious, yes. And what's more, what is even more important, you see estimates of statistics, and you may see that anomalous signal is similar to error systematic, so it would be difficult to extract that. We have to do something better. And you see that on the beamline. So you can correct things on the beamline. Now, there are many papers which are saying that, okay, our methodology is wrong, we should do something better. And for instance, there was a paper, my friend was one of the authors, and they wrote that we know that in some X-ray structure of the ligand-free, it was SARS-related structures obtained at 100 Kelvin, this potentially crucial water molecule is absent. Yes, and they suggest that, look, we should abandon cryocooling, we should collect data in room temperature using millions of crystals. Now, if we will look at the PDB, but not only in the model, but if we will look in the map, this catalytic water is there. But you have to look in the map. Now, we had some talks of the people who are looking at structure. Did any of you look at the map? Whether map agree with the model? I do not know, at least my friends who are not crystallographers, they ever look at the map. And I review some papers which had to be rejected just because they took the model, and model in this particular places was bogus. Now we have PDB redo. They are using newest tools to re-refine the structure. And structure is better, but still water was not there. Yes, why? Because PDB redo does not change anything in the model. It makes better refinement. So guys, we should be careful. And especially if we are in a situation like we have now, and this is the COVID-19 bioreproducibility.org. This is because people deposited structure very fast without publication. What we have done, we put all SARS-CoV-2 related structure, and we try to find errors in that. Yes, what was suboptimal, and it result was many papers published in short time. Many of them were on the journal cover, and it was fantastic collaboration, I have to say, of several institutions. But what I would like to tell you that this is resolution, this is quality of the structure, but quality in general sense. Not always it's important to have, but we divided all possible corrections into minimal, moderate, and significant. And what is significant are biologically relevant corrections and wrongly modeled or misidentified primary ligands. Good are free, it's not significant error. Yes, there is one thing which may be minimal and significant. It's non-standardized cell placement. There are many structures which were very similar, but they put in different place of the unit cell. And for biologists, they are very difficult to compare. Yes, for crystallographers, no, we can do that very easy. But we have to realize one thing, that our consumer, PDB consumer, is not another crystallographer. This was true 50 years ago. It's biologist. And biologist not necessarily is structural biologist, and even structural biologist is not necessarily crystallographer. So we have to do everything for them. And I'm not going to go into details of that. And before we were prepared to that, because before we analyzed most important structures which were dealing with antibiotic resistance. And this is a close look into structural models and primary ligands of metallo-beta-lactamases. And again, it was beautiful collaboration. And we always, when we found the errors in structures, we contact authors and ask them to redeposit structure. And the end of that was that we were invited to write a paper about refinement. We are not experts of the refinement. Yes, we never wrote refinement program, but we just use the program on really looking on things which are important. This was possible, and I will come back to HKL. This was possible because we, HKL 3000, by click of the button, produced the error report, the structure quality report during structure correction or during structure determination. And here you have correlation coefficient between MAP and residues, and mostly ligands. Here, there are many waters which are unjustified. Here are ligands, and you can look at the map, how map covered the ligand, and now you may even go into outside and do that in, you can spin the ligand and you can find the really good reality. So now, I would like to say the statement by Stephen Hawking. The greatest enemy of knowledge is not ignorance, but the illusion of knowledge. And so quite often people really have illusion of knowledge. And this is something which is partly related, because HKL 3000 is really driven by the database. And Washington Post had messy, incomplete US data, Hubble's pandemic response. This is absolutely true. We have analyzed the data, and in many places, data do not have any sense. There are some counties where total number of cases which happened in this counties is going down. Total, not per day or not per week. Total is going down. How they did that, I mean, I know, but it's not the place to do that. And what is the future of structural biology is problem-solving approach, integration of structure and medical data. And this is our example of that. What is interesting, that this structure, this is complex of albumin with dexamethasone. This was done in 2011 as in structural genomics. We did that, but we never refined. We published many structure and we did many structure, and we're not able to do everything. And we thought in that time that it's not very important. But when we have learned that dexamethasone was the first drug against COVID, we immediately, because we have database, we immediately grab this data, refine, and compare with medical data. What we found that when your level of albumin is high, you have very good chance to survive. And I am one of them who has high albumin level. And this is, again, the beautiful example, which was published where people go through all in spike region. They they tested all possible binding sites and connect that with crystallography and KOEM, and it led to sotrovimab, which was already approved by FDA. And according to doctors in my school, it's very potent, but unfortunately in short supply. And the last thing, this is software. And software is quite often underestimated. Why? Because it's enough to use command CP to copy the software to another place. And people think that software is easy. But look, 737 MAX, yes? After disaster, supervisors said, okay, it's not a problem. This is only software. We will fix that in 2 weeks. It took almost 2 years to fix that.
Vladek, please notice that we are 5 minutes past time, so please try to wrap it up. And
yes, I'm sorry. This is last slide. This is last slide. Almost last slide. You see that this is the beamline. What people are doing, they are looking at the map. Of the map which was produced by the crystal, which is in X-ray. Why? Because they are thinking what to do next. Yes, and this is my message. Do not do everything at the beamline as much as you can. If you are sending crystals, I know that it seems cheaper, but you are not as effective. And I would like to acknowledge many people, especially those who were in the last time. And I would like to acknowledge my lab. It's plus 111. I couldn't put all names. Thank you very much.
Thank you very much, and thank you very much for all of us who stayed until the very end. And because we are past times and there's no time for questions, but and I have to leave. But if anyone wants to stay and continue discussion, please go ahead. Thank you very much again for a very interesting session.
Thank you.
Yeah, thank you, everybody.
And as I said, I can be available on Tuesday, let's say, to answer a question or for this discussion over Zoom, which I may organize.
So thank you, everybody, for attending, and thank you very much, speakers, for taking your time and putting in all the energy into this. We really appreciate it. I hope you all have a great afternoon. Bye-bye.
Bye.
Thank you.
Thank you, Chuck.
Jim.